Thank you very much, Arnold, for agreeing to give a Build a Cell seminar, and you can get started.
Okay. Okay. So this works like this, right? You see my presentation?
Yes.
Okay,
Perfect.
then I'll go. All right. So welcome all to this seminar. I'm very happy to speak to you here. Let's see, I'll just put the audience in the corner there. So today I will talk about the organization of crowding in reconstituted cellular environments. So just to talk a little bit about myself, as I was asked to. So we are based at Utrecht University. So we have been here for a couple of years. Before this, we worked in Germany, my group, and before that in Groningen. So a lot of things that I will show have been geographically spread out over multiple locations in Europe. But it all fits together in one story, fortunately. So organization of crowding in the cytosol, and what does it mean? So we try to understand how what the crowdedness inside cells means, and what it does. But before I go that, I will take a step back and consider why crowding is important and why we need to incorporate it in building a cell. So there's been a lot of debate about what crowding, what life actually means. There's a lot of philosophy books, and I'm certainly not an expert in this topic. But what is clear that there are several aspects that characterize life. Cells grow, they respond, they reproduce, they perform metabolism, compartmentalize, they have heredity, and they carry over genetic information, and they're in homeostasis. They don't fluctuate too much. I'm pretty sure that many of these topics have been treated in this seminar series. Now, that is a lot to do for a cell, and in order to do that, it cramps all these functions into a small volume. So why does a cell do it in a small volume? Because it's energetically more efficient. You need fewer molecules to have a certain concentration. You need fewer protons to have a certain good pH, etc. And also you have a large cell membrane, so you have a lot of interface interactions that you can use to function as a cell. So in principle, if a cell could have the way it wants, it would just pack itself as dense as possible. Now, at some point, of course, things don't move anymore. You get so full that you lose fluidity. Therefore, there is probably an optimal crowding level. And this optimal crowding level, then you would guess like, oh, that should be the same for every cell. But actually, when you look at the density of proteins inside different types of cells, you find that the concentrations is about 50 to 400 milligram per milliliter, depending on the species, who determines it. And so this is a very wide range for something that could have an optimum value. So what does it mean, these concentrations? So for example, E. coli is about 300 milligram per milliliter of crowding. And the average protein is about 4 nanometers in diameter in E. coli. And if you put them on an equal lattice, the average distance between the proteins is about 5 nanometers between the surfaces. So between every protein, another protein fits. Now, this is just in an ideal situation, but of course you have stresses, you have different energy states, and this causes changes in crowding, and things move differently. So, okay, so crowding is quite variable. It's not like a single value, even though it could be optimal. So this density that leads to a crowding effect has been described in various cases before. So it can be described using the depletion force. So here, what you have is you take into account that the crowders are all inert. They don't interact with anything else. Then what happens is, if, for example, this big protein gets close to a surface, like here in the right case, at some point the crowders don't fit in between anymore. And if you have a lot of crowders outside, you get a large pressure difference between the outside and this depletion volume. And this causes the depletion attraction, and this protein, for example, and the surface will have enhanced affinity for each other. Now this increases, of course, with more crowders, but also it's size dependent, because if this crowder will be smaller, it could still go in between, for example. And also if it's larger, it takes up more volume and there will be more steric clash with its neighbors and increasing this pressure. So crowding, you can see it as a depletion force, and it's depending on various parameters. So what does it do to you, and why should we care? Well, one thing it does, it enhances biochemical equilibria, for example, phase separation and oligomerization. And for example, here as an illustration, for example, if you have a nucleoid, crowders cannot fit in between anymore, and they create a pressure difference and compressing this nucleoid as the outside is highly crowded, and these crowders themselves also need more space. Similarly, with protein phase separation, crowders can't fit in between anymore. They create a pressure on the condensate and making the whole thing more condensed. And this goes beyond the simple cartoons that I borrowed from this paper, but also you see it a lot in vitro. For example, many phase separation experiments are done by the addition of crowders such as dextran, 10%. And then, for example, this poly-Q-containing protein does not phase separate, but as soon as you add dextran, it starts to form droplets at very high protein concentration, by the way. Similarly, you see this often in cells. For example, Wink1 has been claimed to be sensitive to macromolecular crowding in cells, where when you place the cells on an hyperosmotic stress by sorbitol, for example, the cells dehydrate, the crowding inside increases, and this induces phase separation of these Wink proteins. And next to these biochemical equilibria, also diffusion is affected. So can we now predict what macromolecular crowding does to a protein? After a lot of years of research that I will not repeat here, well, that's very challenging, because crowder depends on the crowder inertness. So how much does a crowd interact with its environment? And how much does it interact with other crowders or with the particle that you're trying to follow? Because stickiness will balance this crowding effect. And as you know, many proteins are not that inert. Many proteins do interact non-specifically with its environment. Secondly, also it depends on the concentration of crowders. If you have locally more crowders, there will be higher crowding effect. And for example, in biomolecular condensates, you have a lot of proteins together, but then again, they're also sticky again. So the question is, how does this crowding in a biomolecular condensate, for example, play out? And of course, it depends on the crowder volume and mobility. If a crowder doesn't move, it doesn't interact. And it needs to be big, so it will create crowding in the first place. So this is what I call, these topics is what I call crowder organization. So the crowder organization, how they interact with each other, must be very important. Now, it's not only the crowding that determines the crowding effect, it's also the particle that you look at, for example, here in the bottom. It depends on the size of this volume where crowders can't come. If it's bigger, there will be higher depletion forces. And also if this protein has to interact somewhat to be enhanced by crowding. If it already interacts without crowder, of course, it doesn't have much of an effect. So taking all this into account, the next question is, if we want to build a cell, do we need to incorporate crowding, and is this important or not? Well, I certainly believe that it can be very useful even for topics where you have non-physiological crowding, because if it can enhance phase separation and other effects, it could be very useful for biotechnology effects and also sensing. But what I do is I take it to the other side. I do want to understand how cells work and what builds them and why do they work the way they do. And for that, I want to incorporate crowding inside artificial cells that is actually physiologically relevant. It's almost the same as living cells, so we can learn about what crowding does to proteins, for example, and why is crowding the level that it is. So I posed myself the following challenges. So can we actually incorporate physiological crowding inside artificial cells? And if we even were able to do that, how we know that it is physiologically relevant? How do we know that it is the same as living cells? And to be able to do that, we need to be able to measure crowding inside artificial cells and inside living cells and compare these two systems. So the approach I take to these problems is this busy slide. So what we do is we make new fluorescent probes. For example, we made probes for macromolecular crowding, for ionic strength, for peptide interactions and stickiness, and also for protein self-assembly. We we express these probes directly inside living cells, so we can measure these parameters, for example, in yeast and bacteria and mammalian cells. And then we try to reenact this behavior of living cells inside artificial cells, and we can compare everything with probes. So to be able to measure macromolecular crowding some time ago, we developed a macromolecular crowding sensor. And this macromolecular crowding sensor is based on FRET, on first resonance energy transfer. It's genetically encoded, so the cell can make its own sensor, and we can purify the sensor and put it anywhere we want. So as a donor, we have here in the first version with M-cerulean, and as the acceptor in this version, the M-citrine. But in principle, any fluorescent protein works, and the sensor still works. So without crowding, the sensor takes up a loose conformation, and the distance between the donor and acceptor is very large, and you have a very low FRET efficiency. However, in the presence of macromolecular crowding, crowders are excluded from this region in the middle, and you get the depletion force, and the whole sensor gets compressed together. So the distance between the donor and receptor is small, and you get a high FRET efficiency. Now, there are some important design parameters here. So one is that this sensor was designed in such a way to have very little attraction to crowders, because that would compensate for this crowding effect, as I mentioned earlier. And we did that by incorporating these helices, which are switterionic, so they are not charged. They have no net charge, and this lysine and the glutamate have a ion pairing, so they are very repulsive and don't interact with other charged proteins. So it's hydrophilic. Moreover, it's an alpha helix, so the amide backbone is shielded. And also these linkers are fairly inert, and also the fluorescent proteins don't have a tendency to interact very much. So that's one design element. The other design element is that these helices also increase the size of the sensor, because if it would just be a random coil, it would make double the size. There's only so much that you get bigger, but by taking in these rigid rods, you make the sensor much bigger, so you can get to large volume changes. So this sensor works like a charm, and it still works after having developed it so many years ago. And just to show a bit how it works, so it's really like a polymer. That's how you have to see it. So this is a molecular dynamics simulation done by a group with Patrick Onk in Groningen. And here on the left, you see it, and you see the linker being hyperactive and the fluorescent proteins moving slower. This is actually the same as behind the crowders, but otherwise you can't see it, so we superimposed it here to the side. And the sensor also follows scaling behavior just like polymers do. So I really believe it is mostly a polymer in how it behaves. So we validated the sensor in various different platforms in yeast and mammalian cells and bacteria. So for example, in HEC cells, what you see is under normal growth conditions, you get this ratio. So this is a measure for the FRET. You will see that throughout my presentation. So when this ratio of the acceptor over the donor is lower, you have a lower FRET efficiency and you have a lower crowding. And if it's higher, you have a higher crowding. So then when you add sorbitol, you have an increase in crowding, as you expect. As the cell gets dehydrated or less hydrated, and the density increases. And then when you apply again an osmotic stress, the crowding again goes up. Now, what cells do when they shrink, they recover. They do that by uptake of potassium so they can regain their volume. And that's also what we see over a time frame of 10, 20, 30 minutes, is that the crowding reduces and you can stress them again. So this is expected, and this is what we expected from literature. But when we started to investigate more different cell types, different conditions, we started to get readouts that were not really on what we were expecting. For example, when you take E. coli cells, which we immobilized in microfluidics, so we could follow single cells in time, and what we saw was when we increased the osmolarity by flowing in 300 millimoles of sodium chloride, is that we first saw the expected, after 2 hours, the expected increase in macromolecular crowding. So when you saw an increase in the macromolecular crowding, then the cells recover, and you see a slow decrease in the crowding inside the cells. But the cells don't, but there's an overshoot in the crowding response. The crowding is actually lower when they grow at higher osmolarity, which is interesting, and it's counteracting to what we would expect. But other people with diffusion measurements already earlier showed some discrepancy here. So what we hypothesized at that point, even though we weren't able to really understand why that was happening, we suspect that there was a change in crowd organization. So this is an extreme cartoon, but if the crowders would all move around, you would have higher crowding, but if they would all be organized, and you have less dense spots where the crowding sensor is, you would measure a lower crowding. And this also corresponded to diffusion measurements. So crowder organization could be in effect. And we got more evidence in this direction later when we made spheroblasts out of E. coli cells. So here we use a crowding sensor again, but now we use MEGFP, M-scarlet as donor and acceptor. That also works. And then you see a large increase in the FRET efficiency and the crowding when you make spheroblasts. And the effect is actually very quick because you can just add a little bit of penicillin to the E. coli cells, and within 1 minute, you have a large increase in the crowding. And the cells recover over many minutes, over many hours, in fact. So what's going on here? Because this is too fast for protein synthesis or anything biochemical. And also the volume didn't change in the way that it could be the case. But when we imaged the nucleoid, because that's a main organizer in the cell, we saw large changes. We saw, well, this is actually not the nucleoid. This is actually a stain that also stains the RNA. But what we saw is that without penicillin, you see an organization in the cell. But when you add penicillin, you see a mixing in the cell, similar as spheroblasts. So you see this with this particular stain, but also with DAPI staining, for example, is that the organization in the cell immediately changes when you add just a little bit of penicillin that damages the cell wall. So we hypothesized what could be going on here. So what could be normal growing cells, then most of the protein is separated from the nucleoid, which is fairly condensed, besides the proteins that actually bind the nucleoid. And this would then have comparatively lower crowding effects. Interestingly, the nucleoid is also compressed by macromolecular crowding itself. But in these cases, we see a lot of mixing. So one hypothesis could be that due to the mixing between the crowder and the nucleoid, the crowding effect increases, as for example, the nucleoid takes up more space. There are some alternative explanations why mixing would increase the crowding, but we have not yet figured out what precise the mechanism is in a molecular level between the cell wall damage and the mixing and the crowding itself. This is still ongoing work. So, but it's clear that crowding is not simply a change in volume or protein concentration, but it can also be due to the crowder organization. So to recapitulate, so we can measure crowding in bufferant cells. We have evidence, at least we have hints at least towards that crowding organization changes macromolecular crowding. I would say we still need the direct evidence that this is the case. So the question also comes then, so how do we recapitulate all this active behavior inside artificial cells? So how do we know when we make an artificial cell that we have physiological crowding? And that's the challenging part. So we proposed ourselves the following challenge, and that is to actually do that. So we don't want a simple polymer or protein inside an artificial cell. We did that a lot before, but we wanted to recapitulate this this organizational type, this emerging behavior. So we thought that the best way would be just to take the components of the E. coli cell and put them in an artificial cell. We're not the first to do this. Other authors have done this before. But the crowding never had been determined inside, and it was not clear to us when you would have the crowding as in living cells. So we did this with the classical phase transfer method, where you have single emulsions, which you, by centrifugation, pass through another monolayer of lipids, so you form liposomes. This is a very well-tested method. So when we did this, we actually had a lot of problems with the reproducibility. It was difficult to get crowding. Sometimes it worked, sometimes it didn't work. And it wasn't really clear to us why it didn't work. But when we started to image them, we did saw that there was a lot of aggregation and phase separation of the lysates in there when we incorporated the crowding sensor and we shrank them. Moreover, sometimes cells didn't even shrink. So then we made two technical advances to make this more reproducible and to incorporate macromolecular crowding inside artificial cells with lysates. So the first step that we did to make the crowding better was to immobilize it in a hydrogel, in a low molecular weight hydrogel, agarose hydrogel. So thereby we could follow single artificial cells, single liposomes, because the liposomes are very heterogeneous when you make them with the phase transfer method. So that way we can follow single liposomes and determine their size and estimate the macromolecular crowding and see how much they actually shrink. So that's the first step. The second step is that this having them in a gel allows to put higher salt concentrations on the top and let the salt diffuse in. So you have a very smooth increase in osmotic stress inside the gel. So cells don't have the time to respond to small changes and not drastic changes by 50, 100 millimoles of sodium chloride. Because what we saw also when you shrink them, and this is also known, is that when you shrink liposomes, they form a lot of these nanotubes and stuff on the outside of the excess lipid. It needs to go somewhere. And you need to give the artificial cells time to do that without having gating effects, and also to give the cell lysate itself time to organize. So when we did this, it worked pretty well. And this is the fluorescence of the sensor, of the direct acceptor excitation, which is not dependent on FRET. And you see a nice homogeneous distribution of fluorescence. So we could shrink these artificial cells 5 times more or less, and then you see a linear concomitant increase of the fluorescence of the M scarlet. After 5 times, you see some leveling off. We don't know precisely why this is the case, but at some point you can shrink it more, but the fluorescence doesn't increase so much. This could be due to the fluorescent protein or due to something else, but this is something that we don't know. But on the positive note, we can increase the concentration of lysates quite a lot inside these artificial cells. So when we measured the crowding with the sensor, we indeed saw that the crowding increases quite a lot with shrinkage up to 5 times. So the starting lysate concentration is about 50 to 70 milligram per milliliter of protein in the lysate. So when you don't have any crowder inside the liposomes, of course, the crowding sensor does not show an increase. And interestingly, with BSA, you see also an increase, but it's less increase in crowding. So at the same concentration, lysate seems to be even a better crowder than BSA. So we suspect that this is mostly because BSA is also prone to self-interact. We also see that the vesicles with PSA are easier to shrink, so they have a less pressure inside, which could be due to self-interaction. So now we have this established, so we can increase crowding and we get to high levels. So are these levels relevant or not? And how do small molecules play a role? So to test this, we first played with the role of sucrose. So so here the red line and of the Fred ratio versus the protein content in lysate, which we calibrated from the size of the vesicles. We saw first that the concentration increases from 50 and some vesicles, not all, but there were a few vesicles that went all the way to 300 milligram per milliliter. And we first played with the amount of sucrose because initially we started with sucrose to increase the stability of these artificial cells. So we first removed the sucrose and we could still make these artificial cells. And interestingly, we also saw that the crowding increased without sucrose. However, when we instead of sucrose used trehalose, we saw less crowding increase. This is interesting in a way because also trehalose is, both sucrose and trehalose are compatible solutes. They also are preferentially excluded from the protein surface, and they improve the hydration of proteins. And this effect is strongest, in fact, with trehalose. So this could be that this plays a role and also can also enhance interaction between proteins, which would alter the crowding effect. So, and to confirm that this is really a crowding and a crowding organization effect, we also measured the diffusion of the sensor inside these artificial cells with all these different additives. So we did this with FRAP. So we bleached the acceptor and measured the recovery over time. And then you really see that the absence of sugars also strongly increases the recovery time. So the diffusion is lower, even though the crowding is higher, and that corresponds. So the higher the crowding, the lower the diffusion of these the sensors. So there really seems to be a change in organization of the cytosol due to the presence of sugars. So we tested several other compatible solutes, and most of them did not have such a strong effect. However, ATP did have a strong effect, and ATP is known to interact with proteins. Probably many of you know about the proposed hydrotrope effect of ATP, or at least interaction effect with proteins, and that ATP may solubilize proteins that way. So we added ATP. First, we went to 2.5 millimolar, and then if you shrink it, of course, you get to 10 millimolar, which is the physiological levels. But there we did not see much of an effect. However, when we went to very high ATP concentrations, starting at 10 millimolar, so that means that after shrinkage we have about 40 millimolar, then we see an increase in the macromolecular crowding. So interestingly, ATP also does self-interact, so it could be that this also plays a role here. But at least we're sure that at least very high levels of ATP for sure alter the crowding levels. And also this corresponds again with the diffusion measurements, which are where the diffusion is now lower again than without ATP. Well, besides that, we also tested the effect of both sucrose and ATP to see if they were additive, and they were not. The effect was completely different again, and the crowding was lower. We don't know why this is the case, but I think there are many different effects, and in the end, you're working in a very complex solution with a lot of network effects where it's hard to untangle what precisely does what. But nonetheless, we show that small molecules do affect crowding effects in these complex solutions, and this must play a role in cells as well. So next step is, of course, how does it really correspond to cells? So how close are we to living cells? So in order to do that, we compared the crowding sensor in living cells versus those inside artificial cells. Here on the left, this is the same graph I showed before. This is with and without ATP. And then here on the right, these are the readouts inside E. coli. So here we osmotically stress E. coli and we see an increase in the Fred ratio. And then we see that cells growing at physiological conditions. So just this is in medium. We see that the crowding levels is just as high as inside the artificial cells at about 220 to 300 milligram per milliliter, depending on the small molecule, of course. Yeah, so that means also that we can reach the crowding levels inside E. coli as in the artificial cells, or actually the other way around. We can reach the levels in the artificial cells as the E. coli. However, we cannot reach those crowding values after osmotic stress. I don't know what is missing there, but this is not achievable for us. And we also tested it versus HEC cells. So HEC cells are known to be less crowded than E. coli, and that's also what we see with our crowding sensor. We see a much lower Fred ratio in E. coli, in HEC cells, in mammalian cells. And there, when we compare it to our artificial cells, we are at 50 to 100 milligram per milliliter. And that is also kind of close to what is expected from previous determinations. So it's straightforward to get the crowding levels as in HEC lysate, and also we can get E. coli. So before I go to the next topic, so we can get these crowding as in living cells, but I do have to admit that doing this in liposomes with this phase transfer method, it is challenging. And Milara, who did the work, was very persistent and was able to optimize all the conditions to get this to work. But we wanted to have, at the same time, when we were doing this, we also wanted to have a more robust method, a method that we could always get the same result, and that would also produce the same artificial cells and so on. So we also were working at the same time using with microfluidics to make artificial cells. So our microfluidics artificial cells that we also made crowded, we did this in the following stepwise approach. So we first had the interface of the artificial cells with macromolecular crowders, which we combined with the oil phase containing lipids to make single emulsions. Then these single emulsions stabilized by lipids encounter then the outer phase at the next junction, and you form water oil and water droplets. I guess many of you have seen this before. But now we have crowders inside. So then we collect them and we shrink them with salt. So just with osmotic stress, so we dehydrate them and we increase the macromolecular crowding. Then using, we can remove the oil using flow procedures to form thinner lipid bilayers. So the system looks like this. So this is on the left, you see the microfluidics, you see the two junctions again. Here's the inner phase, here's the oil phase, and here's the outer phase. And here you see the droplets coming out. Here you see the water phase and the oil phase is a separate. The oil phase is lagging behind a bit because of the flow. And when you collect them, they form these nice water oil and water emulsions. We used several types of alcohols, also octanol. So this was inspired by the work of Siddharth Deshpande, who also, I think, presented in this series, and by Kees Dekker, who will present, I think, next year in this series. And we also tested several other octanols, and this is quite robust. They're very monodispersed. They're super stable for, well, as long as we could measure. And as long as you keep them hydrated, they're fine. And so we added crowding inside these artificial cells, and we can add very easily just immediately 500 millimolars of salt. They don't break, they just slowly shrink. I think the oil phase really slows down the shrinkage and also takes up the excess of lipids that you don't have in a normal liposome. So the system is much more robust. So you can controllably shrink these guys you can halve their size by adding salt, and we can add different crowders. And if you calculate after shrinkage how much BSA is in there, you get to 500 to 600 milligram per milliliter of BSA. And this is, well, it doesn't get more dense than this. This is pretty much a solid or a glass of BSA inside. And interestingly, also what we see here is that the shrinkage of these artificial cells strongly depends on the crowder. So with Vical, it's more difficult to shrink them than with BSA. And I think this is, again, because BSA self-interacts. So you need less pressure to compress the whole system. So high crowding. So this is, compared to the liposome work, this is almost a walk in the park. But we want liposomes. We don't want that large oil phase. So we then tried several oil removal methods. And the most simple one was just centrifugation. So in all these cases, we make use of the difference in density between the oil phase and the water phase. So by simply centrifugation, the oil phase stays behind, and you form these liposomes. You can also simply increase the rate of the microfluidics, the flow, and you already have the separation of the oil phase and then the water phase inside the microfluidics, but then you do end up with a lot of oil droplets, and the vesicles themselves also contain still quite some oil. I think the most neat approach is to trap them like we did here, and then the oil phase escapes through this gap. This is very neat, but it's quite challenging to do, so it's not for a daily basis in the lab if you want to study crowding. And if you just need the artificial cells, centrifugation is by far the best method. So then we try to make these super crowded artificial cells with just based on liposomes. So then we shrink the artificial cells again with salt to form these smaller compartments, and simply centrifugation, and you end up with these small and homogeneous liposomes with high macromolecular crowding. So to confirm that we were really measuring crowding, we incorporated macromolecular crowding sensors, and we incorporate macromolecular crowding sensors. And indeed, you see that the macromolecular crowding goes up in them. So depending, you can get all these different densities of in a controllable way that we measure here with the crowding sensor, both before and after centrifugation, to very high BSA concentrations. So to summarize, I've shown you that we can make artificial cells in different ways, water phase transfer, but also microfluidics. We can get very high and tunable macromolecular crowding, especially with microfluidics. But with liposomes, we can reconstitute the macromolecular crowding as we find in living cells with lysates. We have not yet tried this with the microfluidic system. We also see that the macromolecular crowding is not simply milligram per milliliter, which was already known, but we really see that it has a lot of different readouts, both in the cell as in artificial cells, depending on what you add. So small molecules should be able to and can modulate macromolecular crowding, and we also suspect that this can play a role in cells. For example, when we see cells adapting to osmotic stress and they have a different composition of small molecules that alter the interactions between the macromolecular crowders. So with this, I would come to the end of my presentation, and I would especially like to thank my team. And the work I have presented was from Bo Chun on the adaptation of the E. coli, and Theo on the cell wall effect on E. coli, Milara Kalacheva, who did all the work on the reconstitution of the cell lysate, and Andre and Luis, who did the work on the microfluidics, and all the current and former team members and the collaborators and the funding agencies. And finally, I would like to thank you for your kind attention. Thank you.
Thank you. Thank you so much. That was an amazing talk. I really appreciate the microscopy. As someone who struggles with liposome microscopy ourselves, there is quite a bit of questions in chat. Can you see the questions?
Yeah, I will.
If you could read a question out loud before answering, because people watching the recording won't be able to see chat.
Sure, I will. Okay. So the first question is from DBC, and they ask, when you say crowding, do you mean using physiologically identical molecules, or any crowding reagents would achieve the same goal, PEG, for example? Yeah, so it really depends on the molecule. Crowding depends on the shape and the inertness of the molecule. So ideally, we just want to have, like I presented at the end, physiological crowding as cell lysates. But I think if you want to make a sensor and you want to induce phase separation, and you don't, and it doesn't need to be relevant, then yeah, any crowder will do, I guess. I hope that answers your question, because I feel that question also was answered during the talk a bit. The question is from Jajesh Aher. Why sorbitol though? Why not just use sucrose or glucose? Any specific purpose or role in crowding? So sorbitol, I think that question was at the stage of the cells, that where we stress cells with sorbitol. So sorbitol is actually added on the outside of the cells. It doesn't go inside of the cells. So you increase the osmolarity outside the cells, and this dehydrates the cells inside to balance the osmotic pressure inside the cells with outside of the cells. So that can be done with anything as long as it doesn't transport over the membrane or is toxic to the cell. So there's no preference for any. So this also with salts and PEG also is also possible. From Tricia, exciting talk. Do you see problems with your liposome stability when you try crowding inside liposomes because of osmotic pressures? Yes, we definitely do see that. So we have to be careful with how much pressure, how much salt we add outside to the outside. If we add too much, you see, yeah, a lot of different things happen. And just by having a gradient of increasing salt, then you give liposomes the time to respond. If you want it really stable, then an oil phase would be ideal. Then a question from Ksenia. Do you have a hypothesis about why crowding increases protein yield? Is it increasing efficiency of transcription, translation, or both? This is very interesting and important work. Thank you for the talk. Yeah, so crowding increasing protein yield. So I think, so I did not do a transcription translation in here, and the effects we looked at are too short for changes in biochemistry, in protein transcription translation. Crowding does change the rates of transcription and translation, and not in a simple way, in fact, because also diffusion matters. If you increase crowding, you would, in principle, things should interact more and should increase affinity, so you should have faster reactions. But then again, there's also diffusion, which has a counteracting force. So in the end, it's a balance between forces concerning transcription and translation. Then Jajesh says, when you say shrinking of vesicles, did you actually see any specific nanotube formation or budding from the parent liposomes, or is it more just a repacking effect? Yeah, so we do nanotube formation. We do see that in bright field microscopy. We see that the excess of lipid out of the artificial cells. Inside, not so much. So I think that that's mostly it, and for artificial cells. I hope this answers your question. So
I think you answered all the chat questions.
There was just another one just coming in from Daniel.
Yeah.
Does your sensor work also with associative complexation, namely when you have two polymers that phase separate? Yeah, in principle, so if you, for example, you can direct the sensor to different spaces, different spots, and you can have it complex with one condensate. People in literature did this with our sensor. So you can, in principle, measure the different domains in a cell based on phase separation. Yeah, that's possible. You can fuse it, for example, to a protein that goes into condensates. Yeah, the challenge is here, by the way, that even though you can do it, there are some tricks, because on the one hand, you can just measure the Fred, but there is more than intramolecular Fred. There's also intermolecular Fred, and it has a tendency to concentrate in condensates. So it's very tricky business to, when you concentrate all your sensor into one spot, that you don't measure the Fred between the sensors instead of within one sensor. So there is a technical issue there. Per the principle, it should work, yes.
Thank you. I think last call for questions. I just want to say I'm really inspired by this work, because I feel like you're dealing with a problem that a lot of the people in our field don't really appreciate properly. That, the fact that a cytoplasm is not just an aqueous bag of enzymes. It's a highly crowded, highly organized, almost a eutectic phase. And we as the field have not been reconstituting it properly. So I'm really looking forward to see what more comes out of your work, because this is really needed.
Yeah, thank you. I do think, though, if you do transcription translation in lysates and it works, then of course then you already have you're quite far already, I think.
It works, but it's not good enough. Like, we don't have ribogenesis, we don't have the yields that are on the level of what we need for metabolism, and I just have a gut feeling that crowding might be what we need. So
there's a network and a sea of non-specific weak interactions that it's difficult to recreate also. Because we don't have enough nucleoid in here, for example, which is of course a major organizer, and all the RNA, and then we don't have that organized. So there are levels of organization that also depends on the cell fitness. I mean, I guess even in the cell, the transcription translation is not always the same. So
yeah, exactly.
Yeah.
All right. Thank you very much. And thanks everyone for a great discussion. And thank you, Arnold, again for a wonderful talk.
Thank you, Kate. Thank you.
Have a great rest of your Monday.
Thank you. All right.
Thank you.
Bye.
Bye.
Bye.